I am Dr. Khokon K Dutta. My Christian name is David Moon. I'm working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is Mitochondrial translocation of Txnip or TBP-2 or VDUP1 is the most critical event for inflammasome activation and initiation of inflammatory processes. Khokon K Dutta et al. For the first time discovered mitochondrial location and involvement for thyroredoxin binding protein 2, TBP-2, in the rat renal proximal tubular cells in 2005. We also confirmed the cytoplasmic and nuclear locations for TBP-2 for the first time using direct assay method. The report was published in the pathology section of Nature Laboratory Investigation. TBP-2 is known as thyroredoxin interacting protein, Txnip, in humans. It is also known as vitamin D3 upregulated protein 1, VDUP1, as it is the most sensitive and responsive gene under vitamin D3 application or treatment or administration in the human body. We also reported the physical interaction between mitochondrial thyroredoxin 2, TRX2, and TBP2 in vitro. This was also the very first time discovery internationally. TRX2 in normal condition neutralizes free radicals inside the cellular mitochondria. TBP2 inhibits the antioxidant activities of TRX2 by blocking the thiol groups in the active site. It is to mention here that there are many sites inside the cells that generate free radicals, including the mitochondria. But the major part of free radicals are generated inside the mitochondria, which is mostly very toxic and harmful for humans and other species on this planet. The detailed functions and importance of mitochondrial TBP-2 or Txnip or VDUP1 remained unknown for several years. Ranbin Cho et al. Published an article in December 2009 in the Journal of Nature Immunology entitled Thyroredoxin Interacting Protein Links Oxidative Stress to Inflammasome Activation. They reported that Txnip directly interact with NLRP3. Uric acid crystals induce the dissociation of Txnip from thyroredoxin in a reactive oxygen species sensitive manner and allowed it to bind to NLRP3. Txnip deficiency impaired the activation of NLRP3 inflammation and subsequent secretion of interleukin 1 beta. In 2010, G2 Saxena et al. Published an article in the Journal of Biological Chemistry with the title Intracellular Shuttling and Mitochondrial Function of Thyroredoxin Interacting Protein. They found that Txnip is primarily localized inside the nucleus of beta cells in the pancreas and oxidative stress leads the shuttling of Txnip to the mitochondria. Txnip binds with TRX2 inside the mitochondria and reduces the ASK1 binding to TRX2. ASK1 gets phosphorylated and initiates the death of insulin-producing beta cells in the pancreas with cytochrome C release and caspase-3 cleavage. Txnip overexpression and TRX2, but not cytosolic TRX1, silencing mimic these effects. This suggests that loss of insulin-producing beta cells in diabetic patients is caused by the mitochondrial Txnip, mainly inhibiting the antioxidant mechanisms and increasing the oxidative stress. Troy Len et al. Published an article in March 2013 in the Journal of Frontiers in Physiology with the title Txnip Shuttling, Missing Link Between Oxidative Stress and Inflammasome Activation. They described that Txnip shuttling into the mitochondria increases the ROS production and high accumulation of ROS inside the mitochondria. Increased ROS in turn oxidizes mitochondrial DNA also. Increased ROS activates the NLRP3 inflammasome complex and initiates the inflammation in the body. Oxidized mitochondrial DNA can also directly interact and activate the NLRP3 inflammasome complex. They also suggested that inhibition of translocation of Txnip into the mitochondria may work as the great therapeutic approach to deal with disease where unexpected inflammations are involved. They also discussed that when there is ROS in the high enough levels inside the mitochondria, Txnip again translocates from the mitochondria to the cytoplasm and directly interact with NLRP3 inflammasome complexes. In diabetes, inflammation is an important pathological causative for the loss of insulin-producing beta cells in the pancreas. So it can be demanded that translocation of Txnip into the mitochondria also regulates the inflammation in diabetes and other diseases. Takami Sho and Jian Zhu published a review article in the Journal of Biotechnology and Applied Biochemistry in October 2018 with the title Role and Mechanism of ROS Scavengers in Elevating NLRP3-Mediated Inflammation. They made a comprehensive model using current knowledge for NLRP3 inflammasome expression and activation. They showed that signal 1, which includes ROS and NF-kappa B, plays important roles in the regulation of NLRP3 inflammasome expression. And signal 2, which mainly includes mitochondrial Txnip, regulates the activation of NLRP3 inflammasome and subsequent inflammation. They also described that ROS scavengers like antioxidants suppresses NLRP3 inflammasome activation and inflammation. It is to mention here that mitochondrial translocation of Txnip enhances the ROS production and accumulation. But mitochondrial respiratory chain is also good sources for pre-radical production. It is also suspected that the third defense system and respiratory chains may have some sort of crosstalks and involvements. Inflammation is a key regulator of innate immunity in the human body and other species. There are different types of inflammation complexes other than NLRP3. For example, NLRP1, NLRC4, and M2. Mitochondrial Txnip possibly plays important roles in the activation process of all types of inflammations. Michael Karim and his colleagues in the University of California, San Diego, found that elimination of damaged mitochondria removes the chance for NLRP3 inflammation activation. They also found that NLRP3 inflammations are deactivated when damaged mitochondria are eliminated by a cellular recycling process called mitophagy. Tak-Hilam-Ben, Sharmulata Devi et al. Published a research article in March 2012 in the Journal of Diabetes Research entitled Txnip links innate host defense mechanisms to oxidative stress and inflammation in retinal Mueller's glia under chronic hyperglycemia, implications for diabetic retinopathy. They reported that knockdown of Txnip reduces inflammation and gliosis in diabetic retina. Txnip ablation in vitro prevents rod generation, restores ATP level, and autophagic LC3B induction in rat molar cell line. Tak-Hilam-Ben, Sharmulata Devi et al. Also published an article in the Journal of Cell Death and Diseases in May 2017 entitled Txnip regulates mitophagy in retinal Mueller cells under high glucose conditions, implications for diabetic retinopathy. They reported that Txnip is a vital regulator for mitochondrial damage and mitophagy. They also reported that Txnip knockout via Cas9 and Txnip guided RNA prevents the high glucose-induced mitochondrial damage and mitophagy in rat Mueller cell line. From the above discussion, it can be concluded that mitochondrial TBP-2 or Txnip or VDUP1 is a key regulator of ROS production inside the mitochondria, mitochondrial damage, mitophagy, inflammasome activation, and inflammation process in humans and other species. But to make clear, it may be nice to mention here that there exist pathways where inflammation and inflammation is also activated in a Txnip independent way. I would like to wish a very happy, prosperous, and dignified life for you, everybody. Thank you.